Good morning, everybody. I'm Brian Bodner. I'm from the Department of Chemistry and Biochemistry, and I'm going to tell you a little bit about some of the work that we do. Particularly, I'm going to tell you about arsenic. Now, this work falls into the category of metabolomics, which is the study of small molecules in a sample. Now, we're all familiar with arsenic. Um, it's actually a high priority contaminant on the EPA list, and that's because it is present in significant concentrations, essentially spread out around the world. And from these maps, you can see, particularly in the soil in the United States. And so if we look at the map on the left, you can see essentially fairly high concentrations in the soil in various regions. And if you hone in, you can actually see that western Montana is highly impacted with that. If we look at groundwater, there are also regions with high levels of arsenic. In fact, that actually is an issue in the Gallatin Valley. So here is a map showing you a series of wells that have been sampled around Belgrade, Manhattan, and Logan. And these wells shown in red essentially are ones that are above the EPA recommended level of arsenic in drinking water, which is 10 parts per billion, or 10 micrograms per liter. So you might be asking, well, where's this arsenic coming from? Well, some of it is natural. So it's the 20th most abundant element in volcanic activity. And as you know, there is a fair amount of volcanic activity, or historically has been, in our area. It's also used in products. Pesticides and insecticides, which were essentially used all across Montana for years, include arsenic. Fertilizers and poultry growth promoters also have arsenic in it. It's also a result of industrial processes, mining and smelting, and coal. So now, why is arsenic toxic? So first, let's look at the different species of arsenic. So inorganic arsenic is found as arsenite 3 or arsenate, which is arsenate 5. Now, these species are toxic because, one, arsenite reacts with sulfhydryl compounds. So, for example, cysteine residues in amino acids. Arsenate actually is a phosphate analog. So cells can take up arsenate thinking that they are taking up phosphate, which is a required element. Now, there are also methylated species of arsenic, and these are actually formed by cells. These can be microbes, or they can be eukaryotic cells as well. And these methylated species of arsenic are toxic, but it's a much more complicated interaction. So let's look at this microbial transformation of arsenic, because they're important in the speciation and the species found in the environment. So microbial organisms have transporters that can take up arsenate. And in this case, it's actually transporters whose main role is to take up phosphate. There are also transporters that take up arsenite. Now, some microbes have genes, such as this RC, which can convert arsenate to arsenite. And then the arsenite can be exported through transporters such as ArsB. Now, methylation is also another fate. So arsenite, in this showing here, can be methylated by these ArsM or arsenate methylase genes. And then these species can be exported from the cells. And then in the extracellular environment, they can be oxidized and essentially transformed from an arsenite to an arsenate. Microbes also have genes that can oxidize arsenate to arsenite. And we're going to talk a little bit more about the role of those as we move forward. But first, let's look at the periodic table of elements and see if we can understand this, where arsenic falls in the table and what the biological implications for this are. So, as you see here, arsenic is directly under phosphorus in the periodic table of elements. So meaning it has a very similar electronic structure. And this is the reason that cells can actually take up arsenate through the phosphate transporter. It's also led to ideas that arsenate can replace phosphate in certain biomolecules. And so my group, working with Tim McDermott in Land Resources and Environmental Science, and Gejiao Wang, a researcher from China, have done a fair bit of work on understanding essentially microbial interaction with arsenic. And so this is some data showing you, essentially, we're looking at growth curves for cells. So in the presence and absence of arsenate. And what we're seeing here is that as cells grow over time, we're seeing essentially an increase in cellular growth in the presence of arsenate compared to cells without arsenate. And this is the strain GW4. We're also tracking on this plot the concentration of arsenate in the culture media. And so what we're seeing essentially is that as the cells grow, they're taking up arsenate. This is using a different strain of Agrobacterium tumefaciens, strain 5A, and we see a similar profile where the cells grow better when arsenate is present, and they are extracting arsenate from the culture media. So where's this arsenic going? Well, it's going into lipids, carbohydrates, and proteins. So this organism that we're using, Agrobacterium tumefaciens, has actually been isolated from the soils along the Madison River. And this bacteria can transform arsenite into the less toxic form arsenate. It can use arsenite oxidation to generate energy. And as we just showed you, arsenate enhances the growth potential under phosphate stress conditions. So meaning that when the cells are lacking phosphate, we see this enhanced growth in the presence of arsenate. So let's look a little more deeply into the genes involved with this and the protein pathways. So if we look at the regulation of arsenate oxidation in Agrobacterium, what we see is we have AIOX, which is a gene that essentially recognizes arsenite, and that leads to a cellular signal transduction, which essentially then signals the arsenite oxidation genes to turn on. So arsenite oxidation essentially converts arsenite to arsenate. And work that we have done shows that the phosphate stress response controls this arsenite oxidation. Okay? So how does that work? So there's a separate set of membrane proteins involved with sensing of phosphate. So under low phosphate conditions, we essentially get activation of PhoB1, B2, which activates the phosphate stress response, signaling to the cell it needs to uptake phosphate. But this also essentially activates the arsenite oxidation gene response. So even without the presence of arsenite, we see activation of the arsenite oxidation under phosphate stress responses. Arsenate then can actually, this conversion can then be taken into the cell through the phosphate transporters, and we've shown that it can be accumulated into lipids. There's also crosstalk between these, between the phosphate stress response and the arsenite detection, and vice versa. So one of the questions that we're interested in asking is, how does arsenate affect cell metabolism? And so one of the ways to go about this then is to essentially delete individually these different sensor mechanisms. So deleting the phosphate sensor and deleting the arsenite sensors, and then seeing how cells respond. And so we did these experiments where we essentially grew wild-type Agrobacterium tumefaciens in the presence and absence of arsenite under phosphate stress conditions, and we also looked at the delta FOR and delta AIOS mutants. And we did transcriptome analysis and metabolomics analysis. And so we could track a number of cellular functions that were impacted by the presence of arsenite. So just to start, we expected to see differences in the arsenic resistance genes, oxidation, and also phosphate response genes, because we were using conditions essentially to trigger those. We also saw changes in amino acid metabolism, carbohydrate metabolism, copper and iron chelations, and essentially nitrogen and sulfur metabolism. So we saw fairly broad changes in these cells, as well as transport proteins. So let's look a little more closely at what is happening under these conditions that we're using. When we have low phosphate, we're activating essentially the phosphate stress response and this AIO locus. So when we, in the presence, we see activation of this. So then this is essentially leading to activation of the cell is converting arsenite to arsenate. That arsenate then, under low phosphate conditions, is going to be brought into the cell through the phosphate transporters and accumulated into lipids. So there's also interactions between these arsenate signaling pathways and the phosphate stress response and other locuses in the cell. So just to recap our transcriptomics data, it showed that arsenate oxidation is controlled by the phosphate stress response. AIOS with FOR shuts down the phosphate stress response and arsenic resistance when a phosphate analog, arsenate, becomes available. And FOR and AIOS impact multiple cellular functions in addition to arsenic and phosphate responses. So there's really broad implications to the cell in the presence of arsenic under low phosphate conditions. So this is looking at gene regulations. Well, what about the metabolic impact? So what metabolites essentially are impacted by arsenite, and how does that change when we're looking at these mutants that lack the phosphate or the arsenite response systems? So first, let's go back to the toxicity of arsenite. So as I mentioned before, arsenite is toxic because it reacts with sulfhydryl groups. Well, what I'm showing you here are essentially the active sites of a couple of enzymes. So pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. Both of these enzymes have this dihydrolipoamide subunit with two sulfhydryl groups on it. So these can be oxidized by arsenite and essentially inactivating the enzyme. Another enzyme that has a reactive sulfur moieties in it is xanthine oxidase with this molybdenum cofactor that is sensitive to arsenite. So we expected we might see changes in essentially metabolites around these enzymes because they could become inactivated. And if we look at the position of, for example, pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, showing them here in essentially central carbon metabolism in glycolysis and the TCA cycle. Xanthine oxidase is involved with converting hypoxanthine to xanthine. And so we went about this study using LCMS, so liquid chromatography mass spectrometry, and NMR, nuclear magnetic resonance spectroscopy. And both of these techniques are sensitive ways to look at complex mixtures of molecules in solution. And so we're able to look at hundreds to thousands of molecules in a single sample and make quantitative comparisons so we can screen on a wide range what is changing in cells under different conditions. And so this is just showing you some of the data and some of the plots we make. For example, these volcano plots, which allow us to show what species are upregulated or downregulated under different conditions and relate that to their fold change and the p-value or significance of that change. And we map this data onto a metabolic pathway, then we can see essentially, so all of the metabolites shown here in green in this glycolysis, gluconeogenesis, and the TCA cycle, the green ones have increased in abundance, the blue ones are decreased in abundance. And so we can see in this comparison between the wild type cells with and without arsenite, we can see changes where a number of these metabolites in the central carbon metabolism pathways are increased in abundance, rather, while we do see some that are decreased, such as xanthine, which we predicted because xanthine oxidase is inhibited by arsenite. And also we see essentially an increase here, which matches perfectly because the alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate to succinyl-CoA. If this enzyme is inhibited, we expected to see a buildup, essentially, of metabolites behind this enzyme. And the same occurring here at pyruvate dehydrogenase, where essentially these metabolites can no longer flow completely through this pathway, and so they're diverted in other directions. And this is just highlighting those changes that we expected from arsenite metabolism. And we can also correlate this with the gene expression because we had the gene expression data. And so the triangles are showing you essentially what is happening with the genes related to these different pathways. And what we do see is that there's not as strong a correlation with the transcriptome analysis and the metabolite changes as there is if we're looking at, for example, this enzymatic change due to inactivation of these enzymes. And we can do this with also the AIOS mutant and get similar interpretations. And so this can essentially tell us about the impact of arsenite and the influence of these different sensors. So the arsenite sensor has moderate influence, whereas the phosphate sensor has a much bigger influence on these metabolic pathways. So what I've shown you so far is that arsenite exposure can induce global cellular changes in Agrobacterium tumefaciens. We're seeing changes in central carbon metabolism, fundamental nutrient cycling, and transport of solutes through these transporter proteins. FOAR, our phosphate sensor, impacts global functions in arsenate and arsenite conditions, and AIOS has a much smaller impact, and it's really focused on response to arsenate. So the broad implications from this are potential impacts of nutrient cycling in environmental systems, because microbes are the predominant factor essentially determining the speciation of arsenic in the environment, meaning whether it will be arsenite or arsenate. And there is clearly a strong connection between phosphate and arsenate. Now, microbes can convert arsenic between these different forms of the metal, but what happens in humans? So one of the things that has been known is that arsenic-exposed humans show considerable variation in levels of disease. So even after you take into account genetics, lifestyle, routes of exposure, age, and sex, you still can't account for the disease penetrance. And this got us to thinking about the gut microbiome, because we know that microbes can essentially convert these forms of arsenic, and might they be playing a role in essentially arsenic exposure in humans? Because we know already that the microbiome is important for processing, antibiotics, pesticides, air pollutants, and now we're asking the question about metalloids. So let's go back and look at our arsenic genes again. So we have gene transporters involved with taking arsenate into cells. We have arsenite can be uptaken into cells. We have genes that can convert arsenate to arsenite, and we have exporters of arsenite. We've also talked about arsenic methylation. These ArsM genes, which are involved with methylating arsenic, these have kind of a more complex impacts on toxicity, but these methylated forms can be excreted from cells and turned in from arsenite to arsenate. Humans have an arsenate methyltransferase, an ArsM gene. So we are able to do this reaction. This is one of our primary means of detoxifying arsenic. And so we decided to look at some mouse models to see if we could understand, is there an impact of the gut microbiome on the toxicity that an organism experiences from, for example, drinking arsenic in their water. So to make this a simple model, we used a set of mice that have a knockout for this arsenate methyltransferase. So the mice cannot do any conversion. So any conversion of the metal that we're seeing is due to the microbiome. And so when we look at these mice, that they have a knockout of the gene, they have a microbiome, we can see that those mice are completely healthy on 10 parts per million arsenate in their water. If you take out the microbiome, immediately what we see is these, on this percent survival curve, these mice start dying off. And as you increase the concentration of arsenate in the water, the mice die quicker and quicker. So this is really showing us that the microbiome is having an impact on essentially protecting these mice from the arsenate in their water. And so we are now in the process of trying to extend these studies to essentially look at, well, what are the impacts on humans, and can we start to understand the disease penetrance of arsenicosis with respect to the gut microbiome. And so this is work, I'm showing you a lot of work done by Rachel Rawl and Mike Correll, a graduate student in Seth Walk's lab, and my longtime collaborator, Tim McDermott. And here's my research group from the Chemistry and Biochemistry department. And thank you all very much.